Today we'll be talking about transcription and translation. To begin, cells are governed by this chain reaction, DNA to RNA to protein. This can be broken down into two steps. Transcription uses DNA to create RNA, which produces mRNA. Translation is where mRNA creates polypeptides, and this occurs in the ribosomes. Now let's zoom into transcription. Transcription is the process in which RNA copies DNA using its own alphabet sequence, which uses ACGU, while DNA uses ACGT. The mRNA that is formed can escape the nucleus to be used in translation. A key part of transcription is the enzyme RNA polymerase, which uses a template strand of DNA in order to make a strand of mRNA. RNA polymerase builds an mRNA strand in the 3' direction, continuously adding new nucleotides to the end. Transcription is essential to the human body because the mRNA that is formed out of the process goes to the ribosomes and directs protein synthesis. Proteins are essential to the human body for muscles, ligaments, hair, cartilage, as well as many, many other things. So how exactly does RNA do this? Well, here are the 3 stages of transcription. Before transcription even begins, those 2 strands of DNA must unwind near the gene that is being transcribed. This is called the transcription bubble. First, there is initiation. This is a process in which RNA polymerase binds to a part of the DNA called the promoter. A specific type of promoter sequence is the TATA box, made out of adenines and thymines, where DNA is to be read. Every gene has its own promoter, so transcription can take. Once the RNA polymerase is bound to the DNA at the promoter, it separates into 2 strands, one of which is a template strand, which is needed for transcription. The other is called the non-template or coding strand. The place where RNA is first transcribed is called the initiation site. In eukaryotic cells, the main RNA strand cannot directly attach to the promoter, so it needs some help. General transcription factors either make it easier or harder for the RNA polymerase to latch on. They either repress or activate transcription, so the RNA polymerase always binds to the right genes. In order to ensure that the RNA polymerase keeps going in the right direction and doesn't repeat parts of the DNA strand, the DNA-RNA hybrid complex follows closely behind. The DNA-RNA hybrid also ensures that the transcript terminus remains connected. All in all, it ensures that things keep running smoothly during the process of transcription. Second, there's a process called elongation. This RNA polymerase uses a template strand by reading it one base at a time and builds an RNA strand with complementary nucleotides. This chain goes from 5' to 3'. The new strand of RNA is almost identical to the coding strand, except for the fact that RNA uses U for uracil instead of T for thymine. The process is called elongation because the RNA strand keeps getting longer. The third and final step of transcription is called termination, where terminators signal that the end of RNA transcript is complete. A pre-mRNA has been created. However, a pre-mRNA is not quite ready to be used yet. The RNA strand still needs to go through a few more steps in order to be prepared for translation. First, a 5' cap of guanine is added to the beginning of the strand. Next, a 3' tail of adenine is added to the end. These act as protectors for the mRNA. Finally, sequences of introns or junk RNA are removed, leaving only the exons. Sometimes, however, things can go wrong within the DNA and RNA sequences. One specific type of mutation is called point mutation, where a single base in DNA is changed. When a DNA base is changed, then the RNA copies the changed DNA, resulting in the mutation staying. Based on what has been changed, it can have different effects on the resulting amino acid. All of this has been happening in the nucleus. The mRNA, whether it is mutated or not, then moves from the nucleus to the cytoplasm. After termination, transcription is finished, creating an mRNA which is ready to be used in translation. On to part 2, which is translation. Translation is important to the human body for making proteins, but also in order to make sure cells stay alive. In translation, the newly created mRNA is decoded in order for it to make polypeptides. A cell reads the information from the mRNA, which includes the instructions on how proteins are made. These instructions are made using RNA nucleotides, which are A's, C's, G's, and U's, all formed in different groups of 3. These groups of 3 are called codons. There are 64 possible codons, all set on creating certain amino acids. A specific codon, AUG, is called the start codon and signals the beginning of a protein. 3 more codons, called UAA, UAG, and UGA, are stop codons and tell the cell when the polypeptide has been completed. In order for an mRNA to actually be read, tRNAs and ribosomes are necessary. tRNAs read the codons on the mRNAs from the 5' end to the 3' end. Every tRNA has an anticodon which binds to the mRNA through base pairing. Every tRNA also has an amino acid that matches the codons on the mRNA. All of this happens in the cytoplasm, specifically the ribosomes, a structure made out of protein and rRNA, or ribosome RNA. The ribosome has 2 units, the large and a small subunit. It also has slots which the tRNA enter through and bind to the codons, at the same time creating a polypeptide chain by linking amino acids. The slots which the tRNA enters through are the A, P, and E sites. The final product is a polypeptide chain whose amino acid sequence mirrors the sequence of codons in the mRNA. There are also 3 steps to translation. First, there is initiation. All the pieces mentioned above, the tRNA, mRNA, and ribosome, come together and form an initiation complex which begins the process of making new protein. The A site is where the tRNA enters the ribosome so the polypeptide chain can be created. There is elongation. The tRNA amino acids attach to the mRNA codons so the amino acid can bond with other amino acids. The amino acids that have been made all attach to each other, creating a polypeptide chain. This happens at the P site. At the E site, the tRNAs exit the ribosome. Finally, the process of termination begins. It happens when a stop codon enters the A site. These codons are recognized by release factors, which all fit into the P site. These factors add a water molecule to the last amino acid of the chain, which separates them from the tRNA and releases a newly made polypeptide chain. However, like the pre-mRNA of transcription, this polypeptide chain still needs a few edits. Some amino acids can be altered or removed altogether. The new polypeptide chain folds into a 3D structure and joins other polypeptide chains to form a protein. Sometimes the amino acids cannot properly fold and need the assistance of helpers. Just like the mRNA, these polypeptide chains can go wrong. Sometimes the bases are incorrectly inserted or deleted, which can lead to the entire polypeptide chain being messed up. Every single codon would be reading for a different amino acid, which means that the protein that was needed is now completely different. A less disastrous mutation is the base substitution mutation. This change has no outward effect, but leads to termination occurring early, which can badly affect a protein. The processes of transcription and translation are very important to know about, not just because they help us pass our test, but because they are essential to the human body and for every aspect of life. Without them, none of us would exist.